Namaste students. I'm Jaypur Dr. Aziz Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapeeth Women's University, Jaipur. Today I'm going to discuss about the topic cellular reserve material. In this schedule I discussed about some topics related to bacterial structure and their cellular organization and surface structure of bacteria. Talk about the cellular reserve materials. So cellular reserve materials are there or some organs which are important role in storage of food. Storage of food in bacteria. So what are the organs which are involved in storage? First one is the granules. Granules are the organelles which have important role in storing some nutrients. We can also observe these granules by some microscopic methods. And they are the stain. We can stain the bacteria for the observation of these granules, and they can be easily identified. Next one is the inclusion body. Inclusion body is the aggregation of nutrients of the substances, and they are often needed by the cell. So every nutrients will are aggregated in the inclusion bodies. They will be bound in the inclusion bodies. These inclusion bodies may or may not have some membrane bound. They may or may not be membrane bound, and they allow the cell to go for longer periods because if bacteria do not found any kind of nutrients, any kind of food, then in those conditions, these inclusion bodies will avail all the nutrients which is stored in this inclusion body to the bacteria so that the bacteria will survive for a longer times. There are some more cellular reserve materials which also located in the prokaryotic cell and are invariably known as the granular cytoplasmic inclusions. And there are some important organic cellular reserve material which are present in the prokaryotes, which include the polybeta hydroxybutyric acid, glycogen, and starch. So the cellular material are present in the polybeta hydroxybutyric acid, glycogen, and the starch in the three forms. First one is the polybeta hydroxybutyric acid. It is found exclusively in the prokaryotic cell, and invariably they caters as an equivalent of lipoidal content duly they are stored in the eukaryotic cells. So it is like a lipoidal content which is found in the eukaryotic. So, but they are found in the prokaryotic cells, and it is observed in several species of Azotobacter, bacilli, and pseudomonas. Not all the species, only some species of the Azotobacter and pseudomonas. And certain specific organisms such as the purple bacteria has ability to synthesize two types of research material like glycogen and polybeta hydroxybutyrate. Not so these purple bacteria have special features. It will synthesize two both uh, both kind of polybeta hydroxy glycogen. So it is very specific. And what are the roles? So first one is visibility. These organic cellular material are in, found to be deposited almost uniformly, very much within the cytoplasm. They are visible, and they are may not be detected under the microscope, but they are visible only when they are stained. They are visible, but they are visible only when they are stained with dye. cellular content of direct acid, the activity one cell do have three reserve materials present in small quantum in cellular content, which is series culture medium. The availability, these reserve material may sometimes present even up to 50% of the total cellular reserve material which is contained in the databases. So it is highly present inside the body. They are highly present inside the bacterial cells as compared to some another cellular reserve materials. Utility, if we talk about the utility, these reserve materials are fully utilized when some prevailing cells are adequately provided with a suitable source of nitrogen and the growth is resumed and subsequently. So these are utilized when nitrogen is sufficiently utilized. Next is the glycogen and starch. Glycogen and starch are stored in the form of carbon by osmosis. And in the particular instances of beta hydroxylations, it precisely designed a method of neutralizing acidic metabolite. Cyanophine, it is also a copolymer of arginine and aspartic acid. Eukaryotes fail to store particularly the organic nitrogenous material, but the blue-green bacteria is expected to accumulate in nitrogenized material which is terminated the cyanophycin. It invariably represent as much as the 8% of the cellular drive and they may be regarded as copolymer of the arginine and the aspartic acid. So cyanophycin is also a cellular reserve material which is found in some selected type of bacteria. Volutin granules. Volutin granules are definitely present, particularly the intracellular phosphate reserve. They are when they are required, they are designed at the nucleic acid synthesis. They are desired when nucleic acid synthesis is failed, and the eukaryotes will acquire more and more volume, and they may be stained by methylene group. And in fact, these prokaryotic appear as red on being stained with the blue dye. The actual mechanism of these volatility in prokaryotes take place under critical parameters of starvation, which is best known as the sulfate starvation. The volatile granules disappear as soon as the cells adequately made available as sulfur. So they are only synthesized when the sulfur is not present. But when the sulfur is available, they will, these molecules will start disappearing. And subsequently, the phosphate molecule may also be uh, found to be utilized when the molecule is present. But if the phosphate is also there, they will also start disappearing. Sulfur bacteria, purple bacteria are some photoorganic bacteria and some filamentous and non-photosynthetic bacteria are some examples. And these two sulfur bacteria specifically help in the accumulation of the sulfur hydrogen sulfide oxidation. So these are also some examples. Thylakoids. Talk about the thylakoids. These are solely present in the blue-green bacteria and are intimately involved in the phenomenon of the photosynthesis. And there are three structures, namely gas vesicles, chromobium vesicles, and the carboxysomes that are bound by some non-unit membrane and have been reported by photosynthetic organisms. Rips. There are several aquatic eukaryotic essential containing gas vacuoles that are ultimately engaged in counterbalancing the preventing gravitational pull. And these gas vesicles are usually surrounded by a layer of proteins approximately 2 millimeter thick, and these structures possess several bands consisting of regular rows of segments that almost run perpendicular to the axis. Photosynthetic apparatus are also some cellular materials, and the photosynthetic apparatus which are present in the photosynthetic bacteria, and they are usually bound by a series of the cigar-shaped vesicles which are arranged meticulously in a cortical layer, which ultimately underlies the cell membrane. These structures have a widely nearly 50 nanometer length, is vary between the 100 to 150 nanometer, and are delicately enclosed within a single layer membrane. They essentially and invariably contain photosynthetic pigments. Carboxysomes are also some organelles which are the cellular reserve material organelle, and some specific photosynthetic and cumulative organism like algae, purple bacteria, and some thiobacilli, essentially composed of polyherbal structures which are known as the carboxysomes. Carboxysomes contain some enzymes that are involved in the critical fixation of the carbon dioxide, such as the carboxy dismutase, and thus they represent the precise and most probable site of carbon dioxide flow. In the photosynthetic as well as in the cumulative topic they are found. So these carbon dioxide fixation is found in the both photosynthetic and cumulative trophic organisms. So in this lecture I discussed about some organelles which are involved in the cellular reserve material, which include the granules and the inclusion bodies. Similarly, some materials which are reserved include the poly beta hydroxy, butyric acid, glycogen starch, cyanophacin, polylutein, and lips. So these were described in this video. Similarly, some structure like thylakoids, say photosynthetic apparatus, carboxysomes are also discussed in this lecture. So students will be asked that explain different cellular matrices. So you can answer all these things. What are the organelles which are involved in the cellular reserve materials, and which was important organelles are found in the blue green algae, purple bacteria. So these are each uh, thing is explained with some examples here. So I hope you will be able to answer all these questions. So my lecture is over here. If you have any query, you can